So with that, I want to thank everyone again for coming tonight. I know that Monday night at 5:30 isn't ideal for everybody, so I really appreciate you guys being here. I'm very excited to introduce our tonight's speaker. Ben and I have known each other for a very long time. Maybe if you stay to the end of questions, we'll tell you the story of how we first met. It's pretty hysterical. We just went over it again. Anyway, Ben Gahagan is a diadromous fisheries biologist with the Mass Division of Marine Fisheries. He's going to give us a talk tonight on his striped bass research and management. And his talk is titled, They Have Tails, Understanding Striped Bass Movement and Migration to Improve Management. So with that, Ben, take it away.
Thanks, Sarah. And I changed the title without telling you, so I apologize.
Way to make me look bad. Thanks, Ben.
No, that's my bad. I look terrible. I look terrible. All right. It's never Sarah's fault. It's my fault. So yeah, thank you very much, Sarah. I'm going to be giving this talk about striped bass. I work at the Division of Marine Fisheries, as Sarah said, and we do a lot of work on striped bass in Massachusetts. I'll go over why we do that work, but I'm guessing a lot of you who are in the room tonight are pretty interested in striped bass as well. I do want to acknowledge my co-authors on this talk who I work with at Division of Marine Fisheries. And also you can see that on the lower right-hand corner of the screen, that sport fish restoration icon. A lot of the funding for this project came from sport fish restoration funds, and those are the monies that the state gets from license holders when they buy. It's basically an excise tax on marine fuel and fishing tackle, fishing gear that then goes back to help restore fish populations and manage fish populations. So everybody here, and also a lot of this is paid for by saltwater license money. So hopefully this is something that you find interesting and see it as some of the ways that we use the money that anglers put into the system to give back to the system. And with that, I'll kind of launch into my talk, and please do enter questions in for Sarah or save them for the end of the talk because I love answering questions. Oh, if I can make this work. All right. So I'm going to this is the overview of the talk. We're going to talk about three broad topics here, a background on striped bass for those of you who might not have the most in-depth knowledge of the fish and their ecology. Then we'll talk about what striped bass meant in the past for the Commonwealth and the history of fishery management for them. And then spend a good part of the talk discussing the recent and current research that Mass DMF is doing with striped bass. If anybody's interested in some of this past information, I really can't say enough about this article by my colleague, Gary Nelson. It's really interesting. If you can't find it, email Sarah or email me, find my email online on our striped bass in our Division of Marine Fisheries directory, and I can get this to you. It's really cool article about just Gary spent a long time going through all the historical records and researching everything striped bass and the history of the Commonwealth. Very, very interesting article. So the basics. I love this map. There you can find way cooler, maps of species distributions that are interactive and have pop-ups and everything else these days online. This is kind of hokey and old at this point, and the graphics aren't that good. But what I love about this, and hopefully, usually everybody can see the cursors. Hopefully you can see the cursors. You have in orange the native range of striped bass, and then all this stuff in red is where people like striped bass so much they decided as we went across America. Manifest destiny for and all of that and obviously not the greatest thing, but we put striped bass everywhere we went for the most part because we just love the fish that much. So everything in red is introduced range of striped bass, and orange is the native, and we're going to spend all our time tonight talking about the native range, but the red is where we like them so much we put them so everybody can catch them. This is a basic figure, a little bit complicated, showing in blue the amount of spawning stock biomass as fishery managers that's what we look at. It's the poundage of females that's out there in the water, and you can see over the last 40 years, it was very low in the early 80s. It rose to very high levels from 2000 to 2010 or so, and then it's been coming back down. The red bars are how many millions of young fish are recruiting into the fishery based on a couple of surveys that are done. And then these two blue bars are just management levels. I'm not going to spend a whole lot of time talking about, but you basically do not want to see what we've seen in the recent past, which is our population below these target and threshold numbers for spawning stock biomass. but that's where we are right now. So, as I said before, striped bass, everybody loves them. They bring families together. They look really good as a trophy fish, especially if you put your hands as far towards the camera as you can. And they're also pretty tasty. A lot of people love to bring them home for dinner. They're a very interesting fish. I am a diadromous fish biologist, and diadromous fish are those fish that need to go between freshwater and saltwater to complete their life cycle. And that encapsulates anadromous fish and catadromous fish. Striped bass and most of our other migratory fish here in New England are anadromous. So they're spending most of their life in saltwater, but returning to their freshwater systems, typically the system they were born in, to spawn. So currently, striped bass are, as most anadromous fish, are spring spawners. They're currently found in the largest rivers and estuaries along the east coast of the United States. They probably historically were in many more systems spawning, but we've lost a lot of that diversity of populations. Their young tend to remain in the estuaries for a year or two, at least, if not longer, and they grow rapidly. It's a good environment for them to get food, but escape predation from very large fish. They display something that's called partial migration. They have groups of fish within each population. Some stick around for 1 year, some might stick around for 4 years in their natal estuary, but the other parts of that population, other contingents of that population, move out enjoying what is called the coastal stock. So they swim out into the ocean from their natal estuary, and we call it a stock, and we manage it that way, and I'll get more into this later, but it's really a mixture of fish from all these different spawning populations on the East Coast. So here's the annual migration that a spawning fish, or maybe even a pre-spawning fish, but one that's joined the coastal stock might make. Where in the spring, it leaves their spawning rivers. They migrate north along the coast. In summer, they spend typically in New England in foraging and eating and getting fat and happy. In the fall, they turn right back around and they do it again, migrating south, and they overwinter offshore in the southern mid-Atlantic, where the water temperatures are a little bit warmer. So, and then finally in late winter, they'll enter their spawning rivers. And this is kind of the diagram that people see in textbooks for a really long time. And I'm going to spend a lot of the talk later on talking about acoustic telemetry. And the really cool thing about acoustic telemetry is we can go through these figures I just showed you and turn it into this. So we tagged a couple hundred striped bass, and the size of the dot right now, you can see weekly time lapses. The size of the dots are how many fish were detected on a receiver during that week. So right now we're in August, and like I said, all the fish are in Massachusetts, but now we get to September and October. Fish are coming out of Massachusetts and going back down in November along the coast, overwintering here in the southern mid-Atlantic bight, going up into the spawning estuaries starting in late February, early March, April, and in all the spawning estuaries now. Mother's Day, as a lot of people in Massachusetts know, is a good time to start fishing. They're in Massachusetts. Getting in the summer, all back in Massachusetts, August, September, turn around, start heading back south and going back down to coastal Maryland, Virginia, Delaware. So now with these tools we have now, we can actually make that whole figure from a textbook come alive, which is really, really cool. So I has a little bit of a sneak peek to the rest of the talk, but getting back to why striped bass are important for Massachusetts. They're very important for Massachusetts. I'm guessing a lot of people here are on the talk because they like striped bass. On the left here in the pie chart, you can see the contribution by weight to recreational fisheries, and it's almost a quarter of total weight of all the fish caught in Massachusetts. This is from 2019. I need to get a little bit more current landings, but this is very typical year to year. While it's not one of the most important commercial fisheries, it does make a small slice of more than small slice of our commercial landings by value. And we also try and maintain it as an entryway for commercial people who want a commercial fish. So it's still an open access fishery. We do have control dates on that, meaning we typically every 10 years we set a new date where if you have not gotten in by then, if we decide to close it. So right now, I believe it was the last control date was last winter. So if you were to buy a commercial license for striped bass this winter, or next year, if we went back to the control date, you could get kicked out of the fishery. But they tend to reset those every 5 to 10 years. But right now it's still an open fishery. Anybody who wants to fish for commercial striped bass in Massachusetts can, or if they're even non-residents are allowed to buy a commercial permit. The interesting thing is not only are striped bass important to Massachusetts, Massachusetts is also very important to striped bass. While these are migratory fish, as you saw from that figure, a lot of them spend their time in Massachusetts. And what that leads to is that most years, Massachusetts is either first or second in recreational landings. This is actually a number of trips. This is millions of trips. Here's the landings. So, and this is 2019 data as well. So in 2019, Massachusetts led all the eastern states in trips, so effort in landings, and then we were third in commercial landings. And that's a quota-based system that has not changed for decades. We are always third in commercial landings behind Maryland and Virginia. But the point to take home here is that Massachusetts are an important part of our fisheries and our culture, but because we have so many people fishing for striped bass in Massachusetts, we exert a large degree of influence on that coastal migratory stock. So Massachusetts is also very important to what happens with stripers. Let's dip into the past for a little bit. This is a very old piece of text describing the abundance that used to exist of striped bass in coastal Massachusetts. Down in Nantucket, y'all don't quite have the tide range that we have in Bass Bay, where we're talking 6 to 10 feet, depending where you are and what the moon phase is. And here this man is describing how basically settlers were putting seine nets across tidal creeks at high tide, and then at low tide, when it emptied out, they'd kill 2,000 to 3,000 fish at a set, which they needed for food. But as you might imagine, that's a pretty lethal way of fishing, and it kills a lot of fish. And that showed when you looked at the legislative history of fishing for striped bass in Massachusetts. And so some of the earliest laws that we have in Massachusetts had to do with regulating fisheries. So this bar chart you can see is a stacked bar chart. So the number of legislative statutes is on the left, and a timeline across 240 years is on the bottom, on the x-axis. And you can see black parts of the bars are for legislative action regarding weirs, and then the white part is for seines. And you can see that from 1700 till 1780 or so, there wasn't that much. But you started to see in the historical record that there were localities that were getting overfished and losing their striped bass around 1800. And you start to see an uptick in 1800 of the number in Massachusetts, the number of legislative statutes regarding weirs and seines. And that just progressed for the next century. And not surprisingly, that was because a lot of people were noticing that a lot of their striped bass were no longer as abundant as they once were. So while the number of statutes was going up, it was not quite doing the job of preventing overfishing of striped bass, and striped bass were going down. I'm going to take a brief break. We're going to go from talking about legislative statutes and talk about actual abundance. And getting into the 1900s, we can recreate a good record of our commercial landings. So we're going to start using that as our proxy for abundance now. And what you can see is there was a real rebound, and that was both because they stopped, as that stop sign denotes, we stopped allowing seine and weir fisheries for these fish largely. And then we had stable recreations. There was really good recruitment, favorable environmental conditions for young striped bass in the Chesapeake Bay and the Hudson River, Delaware River, a few other places for about 20 years. That led to a real resurgence of striped bass on the East Coast, and a real resurgence in landings, as you can see, going up to nearly 1.4 million pounds per year. The top was 1.38 million pounds in 1973. However, there was still a very low size limit for fish during this period, and there was a lot of fishing. So striped bass populations and landings were going up. It was not uncommon, to find pictures in people's attics. Maybe some of y'all's fathers or grandfathers or grandmothers or grandmothers have pictures of like this, of just a pegboard with large 40 to 50 pound striped bass from a day's fishing. This is the era that kind of settled Cuddy Hunk as a legendary striped bass fishing location. Some of the other places in the Cape and Islands really earned their reputations during this period. However, that kind of fishing and that kind of pressure without adequate regulations led to the eventual crash that we're used to. And beginning in the 1980s, they more or less closed the commercial and recreational fisheries for striped bass. They went under what's called the Atlantic States Marine Fishery Commission management. So this is an interstate compact of fishing that regulates fisheries in state waters. And one of the other things that happened during this time period is the federal government, to help promote recovery of striped bass, forbade fishing for striped bass in federal waters, so further than 3 miles from land. And that's still a regulation that's in force to this day. So, and I mean, I will point out that that helped recover the stock. I skipped over that, but that did help recover the stock back to the levels we're seeing now. And striped bass are largely considered to be a management success story, and one of the few ones we have here on the East Coast. I do want to pause now and talk a little bit about fisheries management, because I find that a lot of people, it's pretty, it's a hard to understand subject. There's a lot of misconceptions about how it works. There's a lot of misconceptions about why things get done. And I always feel like it's a good opportunity when I talk to the public like this to briefly go over how fisheries management works. So all of these resources are what's called public trust resources. And this is a doctrine that's been with us for a long, long time in Western civilization, going back to the ancient Greeks. In the United States, it's the reason that wildlife and fish are not owned by the crown. You know, while there was the idea that the crown would hold it in public trust under the Magna Carta, like here in the United States, we said, no, everybody's gonna own it. We're gonna manage it for everybody. So there's a long legislative history in the United States of fisheries, wildlife belonging to the general public and being managed in their trust. So that idea is that somebody like myself, who is a fisheries biologist, helps manage and create things to better manage these resources that are held for the benefit of people, which means that really it's everybody's fish. You know, it's everybody's deer, it's everybody's duck. These are all resources that we should all have access to. And it's my job and everybody I work with job to help have that access be there and to be better in the future for future generations of Americans. So for fisheries, the way that works is that there's different branches of information coming in. Those include management, science, and policy. These are the 4 main objectives you can read on the screen of the Atlantic States Marine Fisheries Commission. They're pretty wide ranging, but the idea is to have plans, to provide science so that we can get these plans. And we want to protect fish, protect their habitat, protect ecosystem health. And objective 1 is rebuilding, maintaining, and fairly allocating and promoting fisheries on the Atlantic coast. For diadromous fish, that means a variety of things. For fish like shad and river herring, there's not a lot of science and management happening. Eel is very difficult because they're the opposite. They're very hard to manage. Sturgeon are an endangered species now. Really, American shad has not been petitioned for endangered species listing. Eel has been petitioned twice and not even proceeded to full investigation. River herring's been petitioned twice, and both times there's been a listing determination. Sturgeon are listed. So striped bass really are one of the few diadromous fish that are not in significant peril on the East Coast right now. And I just want to point that out. It's a life history that was very successful for a very long time, but is not very well suited for the Anthropocene era, where we have cut off a lot of rivers through dams and done a lot of other things to fish habitat. So here's our objectives. This is kind of the umbrella of how the organization works. I'm not going to spend too much time on this, but there's stock assessment subcommittees, technical committees, all these different panels that are funneling up to where the rules are made, which a lot of times actually that management boards and sections here, this orange box, that's where many of the rules are made. But advisory panels are citizens. There's technical committees of biologists like myself, and then there's more biologists from outside state agencies and federal agencies that are doing plan review teams and development teams. So there's a lot of information coming in, and what I want to really get people to understand, because a lot of times I feel like when people are discussing fisheries, they think these are just like, the scientists have it wrong, and scientists have it wrong. That's not necessarily always true. And I'm not trying to get out of being at blame for something. I'm just saying that this is a process that is science informed and not science based. And the important takeaway I want people to have here is that your voice can and should matter. So whether you're an avid angler or not, this is still, like I said, a resource that we're holding in public trust for everybody. And if you have opinions on these things, then the only way to get them incorporated into the management process is to let people know. So find ways to be involved, whether that's an advisory panel, talking to your local representatives, senators, everything else, working with groups that lobby for changes. Like, it's your resource. Be involved and try and get the outcome that you think would be best. So I'll get off my high horse now, or my hobby horse, and get back to what I was saying after I just said this is the source for the talk title, but it's a value-based system. It's not just a scientific system. I think that's really important for people to realize. So I'm going to start talking about some of this acoustic telemetry research. Most recently, this is a project that morphed into a PhD project for me while I'm working. I've been pursuing that at University of Massachusetts Amherst. And this all started with some hallway conversations between myself and my colleagues, talking about some really acute harvest hotspots in Massachusetts waters and the teens. This is a figure showing the commercial landings and number of bass for the four counties in Massachusetts. Y'all in Nantucket who striped bass fish are well aware of this. For a long time, it was really heavily favored towards Barnstable County and Dukes County, which is not on here because it's just not that much. But and that was only growing in the 2000s. Did not need a figure like this to know that, because like I said, folks who fish out in Nantucket probably participated in these fisheries and saw it out there, that it was pretty much a race to fish and turf warfare and boats. You know, a lot of bad stories came out of this about guys getting upset with each other, boats colliding. It really was a very intense fishery off of Cape Cod and around Cape Cod in Nantucket Sound waters. So we had to wonder, with this intense amount of fishing that was going on in really discrete spaces and times, was there a population level effect that might be happening to one of those spawning populations? Could there be one population that hung out off Cape Cod more than another? You know, could we be having these big level effects given how many fish get caught in Massachusetts that otherwise we wouldn't know about? And we wanted to design a study that would get at that. Because we're crippled by this management ambiguity we've always had with striped bass, is that we have all these spawning rivers, but we just manage them as these coastal stock, as I was talking about earlier. So even though we know they're all separate populations, we just call them one big striper pot. So for our research, we wanted to provide information on stock composition of aggregations, examine the year-to-year stability of these aggregations, and then look if there's any differences in mortality on them by their natal origin, migration route, where they spend the summer. And we decided to use acoustic telemetry and genetic stock identification for this. I'm not going to get into the genetic stock identification today. I can't wait to get into that part of the project, and I'm actually hopefully getting my data in the next month. I just got an email last week that it's all at this massive genomics institute getting run right now, but everything's been extracted. It's very exciting. I'm just going to talk about the acoustic telemetry, which is also really, really cool. So we broke our study out into 3 discrete areas, and we tagged bass in Boston Harbor, in the Elizabeth Island, so in Buzzards Bay and Vineyard Sound, and then off of the Nantucket Sound side of Cape Cod, the backside of Cape Cod. And we tagged 259 bass in these 3 areas over 2 years. On the figure, you can see how many fish per year we tagged in each spot. We broke fairly evenly these fish into the size regulation bins that occurred at the time. So that was sublegal fish were under 28 inches. You could keep a fish, a recreational fish at this time was 28 to 35 inches. And then from 35 on up was commercial. But if you were fishing recreationally, you could keep anything above 28 at the time that we did this study. Now that's different, there's a slot. But at that time you could keep anything over 28. and then commercial fishermen could only keep fish 35 inches and up. We surgically implanted these tags. They're fairly large tags. They're about the size of a pinky or so. And they have a 7-year battery life, which is really great. That's a long time to be able to follow a fish. We then put out receiver arrays across Massachusetts. So you can see red dots and blue dots in this map. The red dots were the gates that we made especially for this study. And then the blue dots are great white shark dedicated receivers put out by Greg Skomel and the White Shark Conservancy folks. So there was a lot, really it's just out of the New Bedford office. There are some in the rivers that are not for great white sharks. That's probably the only place that we have not seen great white sharks on a receiver we put out in Massachusetts, but those are not for great white sharks. So you can see we gated off the Elizabeth Islands and Martha's Vineyard, Buzzards Bay. We have a gate in Cape Cod Canal, off of Provincetown, and then our Boston Harbor area, and then finally off of Cape Ann and off in Gloucester. You can see these schematics, and there's a lot of equipment. There's a lot of boat time, running around doing these receivers all the time. But it was also a really great time to be doing this research because this is a map of all the other people using this technology. So we can tag fish in Massachusetts with these tags, and these are all the other receivers out there that might pick up our fish. So we work through the Atlantic Cooperative Telemetry Network. It's a bunch of other researchers. We all share the data with each other so we know where we're going. It really boosts the power of our studies. And importantly for us, we had all the major spawning rivers covered during the period of our study. So the Hudson River, Delaware River, and Chesapeake Bay. And this allows us to do some really interesting things. So we can look at the spawning connectivity of the fish we tagged in different places in Massachusetts where they ended up going to spawn. This is called a chord diagram. It's a less common figure type of figure, but I think it's really great for portraying this kind of things. And basically every little sliver of this chord diagram is one fish. So this is actually, this brown one here is one fish that we tagged in Boston Harbor. And then in the following winter, we detected in the Delaware River. Here's another single fish that went to the Delaware River from the fish we tagged off Cape Cod. This red is for the Chesapeake Bay and the green for the Hudson River. And you can see overall, we saw a lot more fish going to the Hudson River of the fish we tagged in 2015. This is where they went in 2016 to spawn than the other groups. And there was no real difference depending on where we tagged them and where they went between the three submarine aggregations. We could also look at things like, is there a size effect? And in fact, in this case, there was. So for our smaller fish, or fish less than 28 inches, they were almost entirely Hudson, slightly more proportion for the Chesapeake for the recreational size fish, so fish between 28 and 35. But those 35-inch and larger commercial size fish had a much greater proportion coming from almost 50% coming from the Chesapeake. And that was a pattern that kind of repeated itself with the fish we tagged in 2016 and where they went in 2017, where you can see that the Chesapeake fish are greater represented in these larger age classes. We can also get an idea of how much fidelity these fish have to one submarine area. So if you recall, we tagged fish in more discrete areas, right? We tagged in this area off Cape Cod, all through here in Buzzards Bay and Vineyard Sound, and then just in Boston Harbor. But because we have so many other receivers out in Massachusetts between ourselves and Greg Skomel, we're able to talk about a much larger area and talk about how fish might have an aggregation to just a general region of Massachusetts coastline. And this is a chord diagram again, a little bit different this time. So we're saying in it, we let fish tell us, we tried to tag during the dead of summer when fish were already in a spot, but we decided that this first summer where they weren't tagged would be their indication of where they really wanted to be in case we tagged a fish that was on its way somewhere else. So we have Cape Ann and north of there, Mass Bay. The year 2 just means this is the second year at Liberty. So this green whole Mass Bay area, Cape Cod, inside and outside, and southern Mass in year 2. But then in year 3, you can see there was a really high fidelity to where they went back in year 3. So fish that spent their summer in one place in year 2, almost all of them went back in year 3. Very, very high proportion. A little bit of mixing. Most fish that did not go back to where we tagged them ended up going to Cape Cod, as you can see. Cape Cod's receiving a lot of fish, but not giving a lot of fish. Really just one fish here that went and spent its summer in southern Mass. Another way to look at that is to think about bass that we tagged in 2015 that we have 4 years of history on. And of those fish, about 50, 60 of them, 90% of them stayed within the same aggregation area over all 4 years. And only 10% of them went somewhere else in any of those years. So a lot of fidelity to general areas in Massachusetts. We can also look at migration corridors, which is fascinating. Here you can see on the bottom of this figure is the route, and then the stacked bar charts are counts. And again, the colors correspond to the map. So fish that just spent their entire summer in Buzzards Bay are these bluefish. Fish that migrated into Massachusetts waters, going through the Cape Cod Canal and then exited in the fall through the canal again is this stacked bar chart. Then canal in through the canal, out through the Cape, around the Cape in the spring, and then out through the canal in the fall, and then going around the Cape both ways. And you can see there's a variable pattern for Cape Cod tag fish doing these different things involving the Cape, but over 95% of these fish that spend their summers north of the Cape Cod Canal enter through the Cape Cod Canal and exit through the Cape Cod Canal. And that can make a difference for these fish. So in 2017, 40 of the 136 bass that we had tagged that entered Massachusetts waters ended up making their final detection in Massachusetts. They never left, most likely because they were either predated upon, but more likely angled. We do get tag returns, but that does not account for all of our fish. And so you can see again, is a stacked bar chart and by area where these fish made their last detection, number of bass removed, and then where this happened. And the color coding here is for season. Green is spring, red is summer, orange is fall. And you can see that southern Mass and the canal had the largest contributions to those fish removed. What's important here is that this entire fall bar was northern fish emigrating from Massachusetts waters in the fall, leaving to go on their migration and getting basically removed from the Cape Cod Canal by anglers. It's a famous spot to fish in the fall. It's a big deal to go fishing there, but it seems like it may be having disproportional impact on fish that spend their summers north of Cape Cod rather than all fish in Massachusetts. So this is a really interesting piece of information that I'm looking forward to drilling down into a little bit more in the next year, looking to publish results. So this has also been a changing fisheries in a lot of ways. A lot of the historical documents I showed you, we're talking about harvest. These days, and really since the rebound in stocks, this is largely a catch and release fishery. So over 90% of the actual catches of striped bass are now releases. These are people catching a fish, whether it's too small to keep or they just don't want to keep them, end up putting them back in the water after being caught. And what that translates out to is a scenario where, based on one article, one scientific manuscript written in the 90s by somebody who actually ended up becoming the director of marine fisheries, Paul Diadromous, there's a 9% assumption that 9% of fish that are released alive die because of being caught. So there's what we call a post-release mortality of 9% in this fishery across. And it shouldn't be used this way, but the way it's used is across all gears and lure types, bait types, everything. That's not the study that was conducted, but nobody has a better number, so they just apply it across everything. And what that means is there's so much, you got to remember, you go back to that figure I showed you earlier, there's so many fish being caught every year that even though it's only 9%, that's a number that is actually about the same or more as far as the millions of fish getting killed as the actual number of fish being purposely killed. So over half of the quote unquote mortality on striped bass in a given year now is from fish being released in the catch and release fishery. And again, Massachusetts is typically number 1 or 2 in that category of number of fish caught. So, in 2019, we caught nearly 4.5 million striped bass. So that's 450,000 dead fish, roughly, a little bit less. So that's an issue for this fishery right now, where you can see we have declining, as I showed you earlier, we were kind of below this SSB threshold and target, where we want to be recovering fish. We want to see an upward trend of SSB and population numbers right now, not declining. So really, post-release mortality is one of the biggest issues for this fishery right now and for this fish. Given that, most recently, we have done a striped bass post-release mortality study at DMF. Basically all states now, but Massachusetts was an early adopter, started mandating the use of circle hooks in 2020 for fishing for striped bass when using a live or dead bait. And we did that as a proactive conservation measure, hoping it would work. But it works for a lot of species, but we didn't have a lot of information about whether it would work for striped bass. Given that, we did a 2-year acoustic telemetry study trying to estimate the conservation of using circle hooks in the fishery, but we are also hoping to find some potential indicators, some type of idea of like a common angler, like a everyday angler might be able to note down about whether a fish would end up living or dying after release. I'm going to go over some of those results. This is the first time that we're showing real results from this study, so hopefully people understand. Hopefully I communicate them in an understandable way, and people are interested in this. It has been a pretty hot topic among striped bass anglers. So you can see overall all the receivers that we did get detections of our fish on. That was our secondary array. Our primary array for this study was in Salem Sound, just south of Gloucester, up here on the North Shore. And we had a large array gated off all of Salem Sound and had a bunch of receivers inside the sound, hoping to pick up fish. And what we're really interested in is like the immediate history of a fish post-release. So within the 2 weeks after they were released, what happened to that fish? We felt like a 2-week window was appropriate for assessing what actually happened after one event of catching, being caught and released. After that, who knows what's going to happen. We specifically decided to look at the difference between circle and J hooks. We used hooks. We did a survey of charter captains and tackle shops and tried to pick some very popular, commonly used hooks when we did this. And we're using natural baits, so herring, mackerel, menhaden, and either both live and dead. And we did keep track of everything, whether it was live, dead, and all of that, what hook we were using, what size. And we also had all these other data elements in the study, keeping track of all of this for every fish we caught. We also then assigned fish a condition score based on hook locations and the level of trauma to the fish after catching. And this went from 1 to 4, with 1 being a fish that was, hinge hooked right in the corner of the mouth. Like you can see that condition 1 picture of the fish right here. It's hooked right above the hinge. There's no bleeding. Everything looks good. Put the fish back in the water. It swims away immediately with vigor. That's a 1. And then as you go down that scale towards 4, which is just dead, basically, you can see there's more trauma to the fish, more bleeding, hooks in sensitive areas. So that was kind of our overall catch-all factor variable, was this condition factor. After we did all that, we would then put this little tag backpack on the fish. And that's our acoustic transmitter. We didn't want to do surgeries in this case because we don't want to induce any more trauma than we have to. So doing a really quick wire through the back with plates on each side, and that keeps the tag on the fish. And these tags lasted about 3/4 of a year. And they also had a little, the other ones I showed you before just said where the fish was. Like it was just saying, hi, I'm here, a unique ID for each fish. This tag had an accelerometer in it, so we would know exactly how fast a fish was swimming when we got a detection as well. And if they're not swimming, then it would just come across as a 0, and we had an idea if they're just sitting there on the bottom dead. That's how we were determining whether they were alive or not. So after doing quality control on the fish, we tagged over 2 years. We tagged over 350 fish. We had almost 290, 289 tag fish that we could determine whether they survived or died post-tagging. And we found out that the best predictor of post-release mortality amongst all those variables was that condition score. So here you can see in this figure the 4 scores and the probability of mortality. The dot is the average, and then these whiskers are like the 90, they're basically the range of what they could possibly, the probabilities of dying, covering most of the values. So condition score, that we were happy about that. That made sense. It's intuitive. It was capturing a lot of different ideas about hook location and injury. So the fact that that would be a big indicator of mortality made sense. However, it was problematic for us because it's really hard to standardize that. There were basically two people tagging, myself and one other person at DMF, and we would still have discussions like, is that a 2 or a 3? Like, hey, I had a fish that, you know, it was hooked deep, but there wasn't very much blood. Should I call that a 2 or, you know, like, and it took us a while to kind of really standardize what we're doing. So it's hard to then have other people do that. Thankfully, we could use that data we had from the tag fish and go out to all the fish we caught on this study, which was nearly 675 fish, and say, well, what contributes to that condition score? And maybe that's going to be simpler to have people record. And what we found was that condition was best predicted by a combination of hook location, the time it took to unhook the fish, and the water temperature. And this is a little bit complicated of 2 figures, but you can see here different hooking locations across the top on both rows, the condition scores in colors 4, 3, 2, 1, stoplight motif here. And then this is the probability of getting that condition score. And on the top row here, it's unhook time on the bottom. And so you can see a fish that was foul hooked, so outside the mouth, somewhere in the body, had a high probability, if you had a really quick unhooking time, of being a 1 or maybe a 2, and a very small probability of being a 3. But as you went out, more and more probability of being at least a 2, the longer unhooking time, and so on for each type of hooking location. Here's the same thing on the bottom with water temperature. And so you can see hotter temperatures, higher condition scores, and then, you know, and longer unhooking time, higher condition scores. So that's the way the 3 of these things, and obviously kind of from left to right in the columns, have the least traumatic hooking location to the most. And make it even another way to look at this is, again, you have unhook time and water temperature on the bottom here. And then now we're talking about combining the 2. We've got mortality probability linked to unhooking time and water temperature and hook location through that condition score factor. And you can see these dots are the average for each of these hook locations and the unhooking time. And then this is the white line is the range of those unhook times, and and then the alignment, how high or low it is the probability of mortality. So there were some high, really long values for unhooking time for all these things. The average is down here, and you can see that as unhooking time goes up, the probability of mortality now, not condition score, goes up. You have, especially, you know, just an even quick unhooking time for gills, more than half of those fish will die. And as you go up in unhooking time, the more likely probability of death. And the same thing with water temperature. And as far as our other question and our proactive conservation measures, unfortunately in this study, we did not find any difference in hook type, whether that was size of circle hook or the J versus the J hook. We were surprised by this, given the amount of evidence of circle hooks working in other species, and there's even some striped bass specific work that's been done that suggests that circle hooks would help. We're not sure what to make of this. We want to try a few different circle hooks still, and are also trying to understand if it might change with different bait types. We used a lot of live mackerel in the study, and the way that striped bass attack live mackerel may just not have been a great combination. Maybe there's other things like scup, menhaden, other bait. We didn't do a lot of eel fishing with this. We didn't have a lot. We tried. We did not have a lot of luck with it, but maybe those would produce a difference between J and circle hooks. But those are all things we need to follow up on. But for what we did, we didn't find any difference between circle and J hooks. So the really cool part about this is I showed you that water temperature, hook location, unhook time, these really basic parameters that everybody could keep track of, predict mortality. We can do that now. So that for the next 2 years, we're going to be doing a citizen science project along with work done by us to go target fish. But we're really hoping to get as many anglers and charter captains as we can interested in this project And the idea is that we would ask people to record some basic data information. You can see the list here. If you, it says email, stay tuned. It's hopefully going to be striper@mass.gov. We're waiting for approval of that email address, but hopefully in a few weeks, everybody who has any connection to saltwater fishing will see things on social media. I can email it to Sarah if people want. If you get any DMF emails ever or get our quarterly report, you'll see it there. If you email us, we will send you a packet with data sheets, instructions, data sheets, stopwatches, and thermometers, a stopwatch and a thermometer, so you can record all this information. We're going to be giving out hats to everybody who submits a report. And then we're also going to be having, hopefully, monthly prizes for coolers and fit rod and reel combos. We're hoping to get some really cool prizes. We're working on things right now for people who participate. But as you can see, we have a power to predict mortality from getting some basic data. So now if we can do this across a bunch of different lure types, a bunch of different fisheries, a bunch of different water temperatures, we can actually come up with a really solid estimate to replace that 9% that really shouldn't be used for everything, but is. So that's the overall goal here, is to get enough information for everybody, if everybody participates, to replace that 9% with a more robust number and improve management coast-wide. Just really briefly, I want to talk about climate change because it's the reality of the world around us now. The Gulf of Maine is one of, if not the most rapidly warming bodies of water in the world. You can see this map of changing temperatures along the entire east coast of the United States. And that's the reality of where things are right now. Chesapeake, for the last 15 years, has not been producing great recruitment of striped bass. It's long been the breadbasket of striped bass recruitments where we get a lot of our fish. Whether that's a mixture, that's probably a mixture of land use, overpopulation of humans in the watershed. It's up to 17.5 million people now. But it's also a climate change issue with temperatures, rising temperatures in the Chesapeake Bay. Striped bass spawning is most successful and recruitment is most successful on cold, wet springs, and those are not really the norm anymore. And that's affecting where we are as striped bass anglers and lovers right now, is where we have less striped bass, and there's not a lot you can do to change recruitment. We can do things with fisheries and effort, but we can't do much about how many fish are born. So with that reality, I like to talk about climate change to these fish a lot, because right now, this is the figure we talked about how fish spend their year. In 100 years, 150 years, maybe our great grandkids are going to be on the southern end of this distribution rather than northern end. But if we want to have striped bass like we have now in any sorts of number, we need to start thinking about opening up some of these rivers where historically striped bass were present and they're not now, like the St. John in Quebec, the Kennebec and the Kennebec and the Penobscot have both had dam removals, which are helpful, but more dam removals would be better in these large watersheds that could actually produce millions and millions of baby striped bass. The St. Lawrence in Canada has started to have a restored population. But there's a lot of Nova Scotia, New Brunswick populations that are starting to come back, and opening up the rivers where they used to spawn there to their full extent will help a lot. So we need to start thinking about a stepping stone to a place like the St. Lawrence, which actually probably has, as the environment gets better for recruitment there, like the capacity to be the same size as Chesapeake Bay. If we want to have a lot of fish, we need to help them out and kind of put out a path for them to migrate northward, not just sit there and watch the population go down. And I have this last map on the right up because that's Labrador. And 2 of the last 5 years, there have been massive striped bass fish kills in Labrador where they've never been seen before, because fish are going up in the summer and then they're getting caught up there thermally and they're having die-offs in the winter from cold shock. But I mean, the range is moving northwards. There's, guys who are salmon fishing up there, and instead they're just every cast catching 20 to 25 inch striped bass. So it's pretty wild. It's a different world. It's a changing world. And we want to have striped bass like we always have. We're going to have to help them out. So I want to say thank you to everybody. Here's kind of a list of my summary things that I have been talking for quite a while now, so I'm not going to make you keep hearing my voice, but I will say that's the striped bass conservation plate on the lower right-hand side. It's one of the vanity plates the state sells now, starting about 2 years ago, and a portion of the proceeds of that plate go to DMF to help do striped bass and related forage fish research. So if you love striped bass, please get a striped bass conservation plate. And I'm happy to take any questions if there's time. Hopefully I didn't talk too long, Sarah.
Thank you so much, Ben. I know this is the hard part about not being in person, is like you don't get like the clapping accolades. So just imagine all of the clapping and accolades that's happening. Woohoo! Yeah. So thank you so much. I definitely learned a ton. I have some questions. There's a few questions coming in. I'll just say to everybody, you can drop off if you need to, because I know we're getting close to an hour, but I'm happy to stay on to make sure we get through all the questions, as long as Ben is as well, just for a little bit.
I'm the one who talked long. So,
so we had a few questions about the equipment itself. So one question about the tagging, for the bigger, the first tagging, not the second tagging you mentioned, but are they, when you said they were surgically implanted, are like, can anglers tell that they're there, like so that they're released again? Is it like obvious that that the tags are there?
Yeah, totally. So you would never actually see the tag. I would make about 1.5 inch long incision in the belly of the fish, kind of in between. If you laid the fish flat on its back, it would have been like right about here in the belly. And then we did put a plastic tag, like if anybody's ever caught a U.S. Fish and Wildlife Service tag, like a spaghetti tag sticking out. So there's a little mooring disc in there. I'd suture the fish back up, and there'd be like a little round disc that held the little plastic tag coming out. So that did say, that did have information. I still have a few of these fish running around, still getting detections, which is really cool. But it did say, tagged fish, the transmitter inside, call DMF with our phone number on it. That did not prevent a friend of mine who works on a charter boat in Connecticut from not reading the tag, assuming it was a different tag, filleting the fish, throwing the fish and tag over, like acoustic tag overboard, and then like just handing the plastic tag and being like, here, call the number on the tag. And he full well should have known. And so we got this report in from an angler on this charter boat, and my friend was like, do you think we'll get the tag back? I was like, yeah, John works on that boat. We're definitely getting that tag back. We would totally have saved it. I called him. He's like, that was yours? No, I threw that overboard. So, but yes.
Yeah, and then the gulls that eat the guts like fly by the pit and ping the
that would be a lot for a gull to hork down. It might be, and the transmitters really only work in the water. So I mean, I guess it could happen. Yeah, there's a lot of good stories like that. There's a smaller tag that works more like an easy pass where you have to go through an antenna, and a friend of mine was tagging brook trout in the middle of winter when they don't go anywhere. There were brook trout that were going over a 10-foot waterfall, which they can't go over anytime. And they're trying to figure out how this is happening. And they ended up finding the otter scat with the pit tag in it near the river. So an otter had eaten his brook trout and was going up and down the river. They're like, how is this brook trout going up a 10-foot waterfall in January?
Well, I think, any research, there's outliers, but I keep thinking I've heard of these receivers for some of the other shark research that we've heard about before. And I'm always wondering, like, okay, well, if a striper gets eaten by, I don't know, that the food
Totally.
chain piece, we always think about that. So
yeah, you can see differences in behavior. There's some really cool ways to parse that out. And a lot, if it is a concern, now the tag manufacturers make what's called a predation tag, and it has basically a little disc over a sensor on it that breaks down in the presence of gut acids. And then that changes the signal that's being sent out. So the tag basically has a signal that's saying normal, normal, normal. And then if that gets activated, it just starts spitting out, I'm dead. So that that the predator ate your fish.
See, that makes sense. Okay, So let's see. What else we got for questions? All right, so were there cases where a fish was gut hooked and that the hook was left in the fish? So like a leader was cut, basically.
Yep. And you can see with that on hook time, that's actually one of the things, there's always a debate, like, do I try and get the hook out, or do I spend the time and try and or just cut it and just put the fish back in the water? I think that's one of the takeaways of this too, is that, like, there are people who are really good at getting a gut hook out. There's tricks and you can do it pretty quickly. But if you're not good at it, then yeah, cut that leader and put it back in the water and hope for the best. It's better than spending a long time creating more trauma, tearing, blood, bleeding, everything else, and time out of water, trying to get that hook out. If you're not good at it, then just cut that leader and put the fish back in the water.
Yeah. I did mean to say the Gary Nelson's paper that you mentioned, I put a link to it in the chat. It was the PDF was available online. So if you are interested in the Nelson paper that Ben mentioned, I put it in the chat, a link to it in the chat. All right, so let's see. For the 2-year study, are you only interested in fish data in mass waters, or would data from other states be valuable?
Take it from everywhere.
Yep.
We're unsure about what we're going to get, but we'll take it.
All right, and there's a question. You know, a lot of people have said they like your presentation, so just so you know, you get the accolades.
Thank you, folks.
But do you think the increased seal population has impacted the striper fishery in Mass?
Yeah, I mean, it's certainly going to be a factor. I mean, seals we all know they eat striped bass. We see them eating striped bass. I know on the Cape there's people who are fishing for striped bass, and great white sharks and seals eat them off the end of the line. We've had great white sharks up here doing it. I haven't heard quite as many seal interaction stories from this part of the state as Cape Cod. And I think that it totally happens. I think that it is probably a factor on the striped bass behavior and populations around here. I don't know whether it's having a population level effect or not, but I mean, I also work on river herring and shad. And I mean, you want to talk about fish that are the potato chips of the sea. You know, they're getting eaten by everything. And I have a lot of people who work really hard to restore these fish, and then see just cormorants lining up and slurping down, you know, alewife from a fish ladder outside a fish ladder all day. And it's frustrating, and I get that. But at the end of the day, it's hard to kind of parse it all out. But I really come back to a point of saying all these animals are doing their job within the ecosystem, right? That these are all links in the ecosystem, and they're serving a function. At the end of the day, we're the largest controlling force on all these ecosystems. And if you don't think that's true, I don't know what else to say. We are. So yeah, we're making a choice by not harvesting seals, but I don't know if harvesting seals to help striped bass is really the reason you should make that choice. If we decide to change seal protection, which I don't think we ever are. And so again, I don't think it's like a productive path to go down for fisheries management a lot of the time, because it's not something that's going to change. But at the end of the day, they're doing what they do as animals, and that's just part of the ecosystem. And it's usually, I think, a more productive path to figure out how we can modify our behaviors and the amount of harvest we're taking, or the productivity of some of these spawning populations because of barriers to migration, than it is to try and kill seals or remove seals from the ecosystem. Hopefully that makes sense. A little bit of a rambler.
Yeah, no, and actually that reminds me, Ben, I did want to say personally, I really appreciate talking about the regulations and all the things that go into deciding, especially at the state level, what goes into that, and including the research. So right, like with Science Pub, we're like interested in the science, but I think that's hugely important. Just the land management we do here, we talk a lot about our motivations and our goals and our values, and I think that clear distinction you made between the science-informed decisions and science-based decisions, I think is really important. And it seems like a nuance, but it really makes sense when you talk about that, the trust that these are resources that are valuable for everyone. So you have a say in, and like,
yeah,
say it in different ways.
Yeah, and the public say sometimes counts more or less depending on what the fishery is and who's listening. But my hope for fisheries management is that the public always has a lot of input and they're listened to. And the way for that to happen is to have informed, strong, vocal people wanting to be involved. So the more people that speak up for these resources, if they have an opinion on them, the better.
Yeah, and that makes me, we do have 2 more questions, but it made me think too, when you ended with the Citizen Science Project, was just something that we do a lot here. So I'm like, yeah, citizen science. People are interested in like data specific from Nantucket, then contributing that data is important too. So when you have that information, we can help pass that around the island too.
Yeah, if I knew somebody whose husband was a charter captain.
I know, I wonder who. Or like,
I wonder who I'm going to be guilt tripping into doing this project.
Oh yeah, while my husband is actually still technically in the attendee list, he did just text me he has to go coach hockey, so he had to leave a little early. So we're gonna if you leave early, we're just gonna like sign you up for stuff. So Carl's gonna be collecting some data.
There you go, Carl.
I did actually have I had a question about I'm writing down questions as you go and then I forget. All right. Oh, the release number. So you had some of the I can't remember which part of the city it was, but you had some release numbers. So obviously there was the harvest number and then there's the catch and release number. So where are you getting those exact numbers? Obviously the tracking for migration, you have transmitters, and then are you relying on catch reports for all that? And like, because I think the release information, I'm curious how you know what was caught and released.
Yeah, that is a canvassing program that's made up of in-person intercepts. So we hire about, I think in total, about 40 people, seasonal technicians a year to go out to marinas and boat launches and everything else and interview anglers. And every state does that. It's called MRIP, the Marine Recreational Information Program. It is unfortunately much maligned. It's a really difficult thing to estimate harvest and estimate effort and all these other things. There's a lot of people who try to do a really good job with it, but it's one thing to look at a graph like that and be like, those are the numbers, but there are error bars on those numbers.
Right. I guess that's what I was getting at. Like, I was curious about that.
There's error bars on those numbers, and like all programs, you figure out what's working, what's not, and it gets better over time. But then strange things can happen. COVID was a situation where it became very hard for us to hire people and get staff to do person-to-person interactions. Yet at the same time, fishing effort was probably the highest it's been in 20 years because nobody was working.
Right.
But it was very hard to capture, and there's bigger error bars on 2020 than a lot of other years because it was hard to get enough information.
Right. I think that we find that in a lot of studies across taxa, right? Like the 2020 is the weird blip for
it's like a joke, right?
Data.
Everybody's figure 2020.
Right. Okay, so there's last two questions are, did your initial findings vary at all from the 9% cited in the Diodati study?
Diodati
study.
Actually, no, they're just the circle and J hook, which is like a very limited thing, right? So we don't want to make too much of it. But I think it ended up being right in that 9 to 11% range. And so, but again, that's one bait type,
right?
What kind of like one set of treatments with a couple different hooks and a couple different bait types. I grew up, and so Sarah and I met at UConn. I'm from Connecticut originally, and I grew up fishing in Long Island Sound. And I really learning, for the anglers that were still left on the call, like learning how to live line a mackerel was different than using a hickory shad or a bunker or a scup, which I did a lot of scup fishing. It was a lot different. And they just attack. I think everybody who catches a lot of striped bass, they attack different fish different ways and have different strategies. So I would be really interested. I'm interested to see people from Southern New England start doing this and see if the numbers are a little bit different. I think we're also going to look at treble hooks, and I would guess that if that's where you're going to see a really big difference. So maybe in the end, we are looking more at J hooks coming back, maybe not, but not allowing treble hooks. Who knows? That's way conjecture. Please, nobody, I didn't say that. Never mind.
Yeah, no, it's like, once again, preliminary data with that too. 1 year of data doesn't mean, just like, don't start changing it up. You have to get to have a number of data over different years and lots of different things to be able to make.
Yeah, and anything we do, again, it's a value basis. So anything we do here, it'll be science for managers to use. But yeah, I'd be really interested to see what treble hooks showed against circles and J's, and hopefully we get around to seeing those numbers. We'll be doing some of that work ourselves this year, but we're really hoping to see whatever we do surpassed by the information that comes in from anglers and charter captains.
The last question, I think I sort of know the answer, but was about having a receiver closer to Nantucket. So we do have 2 receivers right around the island.
Right. Yeah.
And I think on the west end.
Yeah, I'm trying to think about
I know there's one at Great Point.
Yeah, and those came about more recently. Yeah. So right now in that image, yep.
Yeah, they're newer.
Yeah. So I think that they just came out. I think that they came in. I think there is one. I'm trying to look. Yeah. So there's one right here on Tuckernuck.
And I know there's one at Great Point too.
Yeah. So I think that came along after, certainly after I made this map, but maybe that may not be through 2020. This map I think I made in 2019. So maybe that came around to get fish. But I mean, it is interesting. So the bird in my beard was a nuthatch, for people who are wondering. But that was a picture that was taken somewhere out in here. We were out in federal waters tagging striped bass, different types of tags, but tagging striped bass on the shoals. And that's a place with a lot of striped bass, as everybody knows. But it's a terrible place for acoustic receivers because it's shallow, it's noisy, and there's a lot of moving sand. So you lose your receiver unless you go there a lot. So it's an interesting technology. It's a very powerful technology, but it's hard to, there's some places where there fish are that it's hard to do it in. And we're always inventing new ways to do this.
And I have to say that this is where this map in particular is where I really appreciate these kind of science consortiums. So like all the different researchers that are putting out these receivers for different reasons. Yeah. Share the data to make it usable. I know the same thing happens with birds. Like there's a different receiver set for birds and bats, and like they get shared as well. And I think that's pretty amazing.
Yeah. So I mean, these receivers here off of Delmarva Peninsula, these are BOEM funded receivers. These are for those wind energy areas, trying to understand the impacts, potential impacts to fish and fisheries in those areas. If you did the current map of this, which is in the last 2 years, there's now a large array here for Cape Wind. Mm-hmm. So yeah, there's all the wind energy areas are doing massive acoustic telemetry studies, tagging a lot of fish, seeing what comes through.
Cool.
So yeah, it's a lot of different, but yeah, a lot of it's these like state agencies, academic researchers. If anybody wants to look into that more of this stuff, there is some publicly available information at a group called MATOS, MATOS. If you Google fish telemetry MATOS, you'll find it. And that is ACT is kind of turned into MATOS, and they have some cool maps, and you can see all the different species and projects affiliated with that group.
Cool. Well, I want to thank you again, Ben, for joining us today. And for the last people who are still here, if you love these kind of talks and you want to hear another one, Monday, March 13th, we have our next Linda Loring Nature Foundation Science Pub. I forgot to mention to go to llnf.org, which is our website, has all our events and things you can sign up for, which are mostly free. And so we hope to see you then. And if you do have any questions after the fact, or you're watching the recording later, don't hesitate to email. You can find my contact information again at llnf.org. I'm Sarah Boyce, and I can relay questions to Ben. Thanks again, Ben.
Thank you. It was great. I'm coming back when you do it in the pub.
Yeah. Yes.